This classic image of a chromosome is not actually how a chromosome looks in a cell for the majority of the time. In fact, the chromosome as presented here as a dyad is only like this from the end of S phase in the cell cycle through metaphase and mitosis. In the cell during interphase, the chromosome is considerably less compact. Why? Because we need to use and access the DNA in our cells. And in the condensed chromosome you see here, it's far too compact to be of any use. So while our DNA does not exist as a fully condensed chromosome, the majority of the time our cells still need effective and innovative ways to condense it because each cell has so much of it. Our cells accomplish this by enlisting the help of various proteins that will aid in our endeavor of compaction and organization of our DNA. The complex of DNA and proteins that is formed from this process is called chromatin. It's important to note that this chromatin complex is dynamic, and changes in it have wide-ranging effects on gene expression, as we will see later in a later video. The first step in DNA compaction is winding the DNA double helix around clusters of 8 histone proteins to form a nucleosome. In this step, 146 to 147 bases of DNA are wound around a histone octamer along with a linker region that varies from 20 to 100 base pairs. The overall structure here is thought to represent sort of a beads on a string look, and it shortens the length of the DNA by about sevenfold. These histone proteins are pretty basic overall, but have a high abundance of positively charged amino acids, which will help them bind well to the negatively charged DNA backbone. The histone H1, shown here, is less tightly bound to DNA than the core histones in the octamer, but it's important in regulating the interactions between histones. Additionally, non-histone proteins play a role in the organization and compaction of the chromosome. In the presence of histone H1, nucleosomes associate with each other to form a compact structure that is about 30 nanometers wide. Scientists, being the creative geniuses that they are, deemed this to be called the 30 nanometer fiber. This interaction among nucleosomes shortens the DNA by about sevenfold again. Our most recent evidence for what this structure looks like shows that this interaction takes the form of DNA sort of zigzagging back and forth between histones due to the interaction between the H1 histone proteins. Up to this point, we have shortened the DNA by about 50-fold from the original double helix, but we're not done yet. The 30 nanometer fiber is further shortened through the formation of loop domains. These domains bind together sections of the 30 nanometer fiber and project other sections out as loop domains. The two ways that loop domains are formed are through the CTCF binding factor, which binds repeats of the sequence CCCTC. When two CTCF binding factors interact, they form the loop domain. The other way is using SMC proteins, which form a dimer that wraps around two DNA segments. Lastly, as you can see in the figure, these two can also work in concert, co-localizing to these particular regions. Each of these chromosomes, organized into loop domains, is then localized into a specific region of the nucleus called the chromosome territory. Here, we see on the left the metaphase chromosomes, and on the right the interphase chromosome territories for a chicken. And this wonderfully illustrates the localization of specific chromosomes in specific spots along the inside of the nuclear membrane. So, in the end, our chromosomes are comprised of these loop domains localized to specific areas of the nucleus. The position of the DNA within these loop domains and how large or tightly packed these domains are is going to affect the activity level of that part of the chromatin. We generally classify the different levels of compaction of chromosomes into two categories: euchromatin and heterochromatin. Euchromatin is less tightly packed and thus more transcriptionally active. These are the areas of the 30 nanometer fibers within the loop domains, and these are the places where we will find the gene sequences in our genomes. Heterochromatin is the tightly packed form of chromatin. This will be found in the centromere and telomeres, along with a few other regions along the chromosome. Most heterochromatin is inactive in transcription. Of course, there is nuance to heterochromatin. There are two types of heterochromatin: constitutive and facultative heterochromatin. Constitutive heterochromatin is always compacted, and this is generally where we find our highly repetitive short sequences. This describes regions such as telomeres and the centromere on our chromosomes. Facultative heterochromatin can interconvert between euchromatin and heterochromatin. And this is a useful characteristic and allows for certain cells to turn on and off gene expression for specific genes. We will explore this ability further in a video on regulating gene expression in cells.